Hello everyone, today we're gonna discuss about how to make phylogenetic tree. In this video, we will learn about how to make consensus sequence from nucleotide sequence. After DNA extraction, we go for sequencing and each sample contains two sequences. These sequences are actually in Bioedit format. Bioedit is a software through which we go for further processing. And each sample contains two sequencing, one for forward reaction and one for backward direction. For sequencing, we first confirm our region of sequencing. It must be ITS, LSU or any other region. I have some samples of ITS region like A29. A29 is basically the sample name or tag name. ITS1F is for forward direction and ITS4 is for backward direction. First we open for backward direction in Bioedit format. For this you must have installed Bioedit format in your PC. Just click on reverse sequence and go for it. As we click on the reverse sequence, this format will appear. A trigger on the left side, you can move it or drag it up or down. This graph basically shows your sequence. This graph basically shows your region of sequence, and merging of two lines or collaboration or interconnection of two or more lines means your sequence is not correct. It means you must have some kind of contamination in your product. Like for end, you have seen there is very close interconnection and overlapping of results. First maximize the screen. Go for edit mode and click on sequence, then nucleotide sequence and reverse complement. The sequence go for reverse, click on edit and copy sequence to clipboard like FASTA format. You can't go for Ctrl C or Ctrl V on Bioedit. You must have to go for edit for copying the sequence and to click file for import the sequence. Must remember this. After copying the sequence, open first file, that must be A29 ITS1F, and same go for trigger up and down and see your sequence till end. In the end you will see there is again overlapping of graphs. It means that your sequence is not correct from the last, but we will go for it because it is not so much. Again maximize the screen, go for edit mode and file and import from clipboard. After that we have to cut the motifs. Motifs are basically sequence of nucleotide. We have to cut. Two motifs are basically here like katas. First we have to search for kata, that must be between 0 or 50 almost. Go for it, CATTA kata and find. It is almost, there must be GAT sequence before kata like GAT before kata. If there is no GAT sequence before kata, we will not cut the sequence. Is that clear? Just click on the trigger on the top and press shift from your clipboard and drag the trigger to the left side and just press backspace or delete from your clipboard. Again, second motifs is GAT like GACCT. You can cut the GAT sequence after GAT sequence. It must be almost somewhere between 500 to 600 or 650. Like here we lies after 510. There must be CAT sequence. CAT is for C888T, not for CAT. C888T. When GAT sequence is joining CAT sequence C888T, then you have to cut the second motifs. Otherwise you don't need to cut the second motifs, right? Just click on the trigger at the top and shift from your clipboard and drag the trigger to the right side and press backspace or delete from your clipboard. Cutting of motifs is complete. Now we have to create the consensus sequence. For this, press Ctrl A from your clipboard. As you can see, both BIFs are selected, right? Go for accessory applications and cluster W applications. Click OK and run cluster W. Okay, now go for alignment and create consensus sequence. Consensus sequence is ready. Click on the consensus, go for edit and copy sequence to clipboard FASTA format. Create a Word file. I'm going to name it test and we can paste our consensus sequence here. After that we will learn in our next video. Till that, if you like the video, give a thumbs up, because it motivates me to make more informative video. Make sure to subscribe the channel. Have a good day. Bye bye.